Yeah, we discussed Ismail's talk on mineral dust as a fertilizer of the Southeast Atlantic Ocean. The first thing we discussed was what exactly was his leaching method, because obviously that's really important for how to assess the bioavailability of iron and other nutrients in these samples, and how that might relate to how they are actually bioavailable in the environment. And Ismail said he'd use three different techniques, including seawater, deionized Millicue as an instantaneous water leach, and as well as a weak acid leach to get the labile component. So I guess the maximum amount that might dissolve in the environment. And he said that he did see quite systematic and interesting differences between the behavior of these three leachates and the samples, in particularly collected inland versus coastal sites. We also discussed about the difference between his work on gravel beds and riverbeds, which are two different geomorphic environments for deflating mineral dust in the Namib Desert. And he noted that although he presented the results for the gravel beds, they were collected using aerosol samplers, so they're collecting dust in the air. And it's quite possible that much of that may have been coming from dry riverbeds. And so that's kind of a future research question, I suppose, in this area, is to quantify the importance of these two different environments for deflating dust. That's yet to be answered. Another question that came up was why the dust didn't go further south and was deflected into the Indian section of the Southern Ocean. And Ismail can correct me if I'm wrong, but his answer was it has to do with the fact that the westerly winds become weaker as you move further south. So that tends to direct the dust to that region of the ocean that he'd modeled it being deposited in. And the final question that we had was whether or not he'd investigated the importance of uranium and other radionuclides in the dust, and he has uranium results for these aerosols, and the summary is that he doesn't think that the aerosol deposition is going to be an important component of uranium inputs to this region of the ocean. Thank you.
We were caught up a little bit with past participants, and we also briefly discussed kind of how severe the COVID situation is in Namibia. And also I brought up the point that those of us that are in countries that are privileged enough to have lots of vaccine, that we really, I think, need to pressure our governments to, disperse and distribute as much vaccine as possible in order to, save lives. And that's the only way we're going to really get out of this thing. I mean, to be honest. And so we really need to, I think, continue to push for that as much as we can. Also, we talked a little bit about how difficult it is to culture Thiomargarita, and we haven't had any success with that, but that it's still kind of a worthwhile endeavor to try to do this. And also the difficulties in still trying to figure out exactly how these sedimentary phosphorites form. Even though there are models, as Carl presented, there's still lots of uncertainties. And so we think maybe we have to look at different types of geochemical tracers and so on going forward to see if we can link processes in the modern more rigorously with ancient the formation of phosphorites in the past.
Yeah, so we were discussing about Nicola's presentation. Basically, we first discussed how her research could be easily translated to the Benguela upwelling system, because it's somehow similar to the Iberian margin, and Nicola discussed the differences in primary productivity between these two locations and how a higher primary productivity can buffer this input of alloctonous materials, but at the same time, how it is more hydrodynamic, which could be introducing older material as well. So it's something interesting to look in this region. Then we have this question by Tim, who's always linking things from here and there. and about or what's the role of sulfurization in biomarker signals, especially regarding alkenones, because they also have double bonds. And then finally, we were discussing the influence of depth in some of the signals that Nicola measured in organic matter, like radiocarbon, which is supposed to get older, or less radiocarbon with depth, and also more reworked organic matter with depth, although Nicola could not see that in her results. That's something that we would expect.
We just had a very brief discussion about evidence for sulfur eruptions and sulfidic eruptions in the sediments off the Namibian coast, because this was related to something that we've just a colleague has just published, finally, Kurt, based on the samples that we collected in 2017, I guess it was. I can't remember, but we finally published this paper on molybdenum and uranium isotopes, and it linked nicely with what Jan was talking about. So.
Right. So we were like a group of 5 people, like a small paper, just we discussed a little bit about some remark and how this experiment and setup was, let's say, interesting and amazing for Rida when he was there. And a couple of questions they asked if this experiment was made by day or night. So it was by day. That's what Rida was his answer. Then the other question is about the mixing layer for the oxygen. So that question was interesting too. So to have the anaerobic layer for any kind of experiment, so that he did use this introduction of this kind of bacteria, I think. Then this kind of species, what he used, it's a specific species of Namibian coast.